Hello and welcome to the ZOOMinar today. Today's presentations are going to be by Dr. Rebecca Berlow from University of North Carolina at Chapel Hill and Dr. Hariom Yadav from University of South Florida. He's there to join the panels. And I also would like to welcome Dr. Chi Zhang, colleague of Dr. Berlow, who's going to introduce Dr. Berlow, the speaker one, and we'll go from there. So Chi, take it over.
Yeah. Oh, thanks, Rams, for giving me this honor to introduce the first speaker of today's seminars, Professor Rebecca Berlow, who is my colleague at University of North Carolina, Chapel Hill. So probably if you stay in the MR field for long enough time, you know Rebecca because she actually has been growing her scientific career actually in the MR field, like ever since she was an undergrad student. So just a brief background. So she received her bachelor degree in chemistry from Johns Hopkins University in 2005, right? And at Hopkins, actually, she spent more than 2 years as an undergrad researcher with Professor Joe Tolman. And if anyone has ever used RDCs, you will know Joe actually has been a pioneer in developing RDC techniques for doing, protein structure dynamics and a lot of amazing theoretical work. I grown up with with reading Joe's work. So after receiving her bachelor degree, Rebecca moved to Yale University, and then there she actually joined Pat Lauria's lab and carried out her graduate work in developing and applying AMR technologies to study conformational dynamics of large proteins, and also including a lot of relaxation dispersion, etc. So she actually really has done a lot of amazing work there. And after receiving her PhD at Yale in 2011, right? So and Rebecca moved from East Coast all the way to West Coast. John Peter Wright's lab, also Jim Dyson's lab at Scripps Research Institute as a postdoc fellow, and later become a staff scientist. There, she actually has done absolutely phenomenal work, has established seminal contributions to the field of intrinsic disorder proteins. I think she will talk a lot about those. And also, earlier this year, so Rebecca has started her independent lab as an assistant professor in my department of biochemistry and biophysics at UNC. And here she's continuing exciting research on IDPs. And we're so lucky to have her join us at UNC. And it's great to have her to present in RAMS the MR series. And without further ado, here's Rebecca.
Thank you so much, Chi, for that really kind introduction. It was such a really special moment to have you have my lab neighbor introduce me. And so thank you so much to the organizers also for this opportunity to present in this series. It's really wonderful that so many of these online series have popped up during the time of the pandemic. And I think it's really great that we kept these going because it really enhances our ability to share our science across borders and really in these, difficult times of travel nightmares and things. So today I will be speaking about intrinsically disordered proteins. These are some of my favorite proteins, and it's always a delight to be able to speak about them. This is, of course, the seminar series on the molecular bases of proteinopathies. And, I should include a brief disclaimer that our research is not necessarily focused on proteins that cause diseases, but simply proteins that actually are playing really important roles in perhaps or giving us ideas of how we can use proteins, and particularly intrinsically disordered proteins, to interfere with common diseases. And this will be a theme that we'll address at the end of the talk today. But so as my title implies, our group and the research that I was doing when I was with Peter Wright and Jane Dyson at Scripps Research, and now we're continuing at the University of North Carolina, is really focused on understanding how intrinsically disordered proteins are able to carry out unique roles in the cell compared to folded proteins. And one of the questions we had about disordered proteins really is best illustrated by, I will attempt to move ahead in my slides. One moment. Let's see. And so one of the critical properties of intrinsically disordered proteins is that they're often involved in networks or hubs that involve many, many different binding proteins. And here I've illustrated the transcriptional coactivator proteins, the Krebs binding protein, or as we abbreviate it, CBP, and its paralog P300, which itself is an intrinsically disordered protein. And it includes a range of disordered domains that are shown here as bars, and the globular domains of CBP and P300 are shown as spheres. And what this figure is meant to illustrate is the fact that the globular domains and the folded domains of CBP and P300 are able to interact with a wide range of different disordered domains. And these disordered domains, if you look at the composition of this list, I don't expect you to recognize all of them, but all of these proteins are involved in transcriptional regulation under different cellular contexts. And so a question we had when I started my work in Peter Wright and Jane Sison's lab is simply, how do all of these different proteins compete for binding of a single folded domain of CBP? And so the work I'll be talking about today has focused on the TAS1 domain of CBP, and specifically on this first protein on the list, which is the transcription factor HIF1 alpha. And so just to introduce the biology behind the work that I'll be speaking about today, we're really focused on understanding how the cell adapts to changes in oxygen availability. And so this is a really well-described process. The details on the slide were actually the subject of the 2019 Nobel Prize in Physiology and Medicine, awarded to Bill Kalin, PJ Ratcliffe, and Greg Semenza. But really the process here that we're interested in is understanding how this transcription factor, HIF1 alpha, is able to interact with CBP and P300 to activate transcription of critical adaptive genes. And so under normal oxygen conditions, this pathway is not active, and that's simply controlled by the stability of the HIF1 alpha proteins. So under normoxic conditions, HIF1 alpha is hydroxylated at 3 sites, and these hydroxylation events trigger it for interaction with the VHL E3 ligase, which targets it for degradation by the proteasomal machinery. And it also serves to inhibit a key interaction between the C-terminal disordered domain of HIF1 alpha and the TAS1 domain of CBP and P300. Under hypoxia, hydroxylation is no longer possible due to the deficiency in oxygen, and therefore HIF1 alpha is no longer hydroxylated, it's stabilized, and it's able to translocate to the nucleus, where it forms an active transcriptional complex with its beta subunit as well as CBP and P300 to control expression of key adaptive genes. And these genes are really important for driving many, as we say, cellular adaptive processes. They allow us to survive under conditions of oxygen stress. However, if these genes aren't tightly controlled, they also can lead to a number of deleterious processes or diseases, such as cancer, cardiovascular disease, or as I'll speak about at the end of my presentation today, blinding eye diseases such as macular degeneration and diabetic retinopathy. And so this process really needs to be tightly controlled. And in the event that oxygen is restored to the system, we go back over here to the normoxic situation where HIF1 alpha would be hydroxylated and then it would be degraded. However, if oxygen is not restored, there still needs to be a critical regulatory mechanism for controlling HIF behavior. And one of these mechanisms is actually the formation of a negative feedback regulatory loop between HIF1 alpha and another disordered protein that it encodes the expression of, called CITED2. And what happens is that once CITED2 is available in the cell, it's able to fully sequester all of the CBP and P300. And that serves to shut down the expression of the hypoxia response genes. And we were really interested in the sequestration behavior that had been observed in some early studies that identified CITED2 as a key negative regulator of HIF-mediated transcription. And specifically, we were interested in understanding what the molecular mechanism might be by which CITED2 is able to displace HIF1 alpha from CBP and P300. And simply the reason we were interested in this is because both HIF1 alpha and CITED2 use C-terminal disordered transactivation domains to interact with the TAS1 domain of CBP and P300. So we hypothesized that disorder was really important for regulating this behavior. And so as structural biologists and of course NMR spectroscopists, we were very excited that we could study these complexes by NMR. And this was work that was actually carried out before I joined the Wright Lab at Scripps. But postdocs were able to solve the structures, the solution structures of the HIF1 alpha and CITED2 transactivation domains in complex with the TAS1 domain of CBP and P300. And while I won't spend too much time going over the structural details here, what I will explain is simply that HIF1 alpha, which is shown here in orange, and CITED2, which is shown here superimposed on the structure in blue, occupy a partially overlapping binding site on the surface of TAS1. And so we can see that there are some regions, such as this HIF1 alpha alpha C helix, that occupy isolated regions on the TAS1 surface where there's no CITED2 that would be occupying that same site. But there are other regions where, such as where these HIF1 alpha and CITED2 alpha A regions are partially overlapping and may need to compete. And then there's a key region, we call it the LPXL motif, and this is a conserved binding motif that is found in all HIF1 alpha and CITED2 species identified to date. And this region, or this motif, binds in the same region, on the same surface, and in the same orientation. And so we hypothesized that this LPXL motif, which is LPQL in HIF and LPEL in CITED, may be playing a pretty critical role as a hotspot for competition between HIF1 alpha and CITED2. The other information we had was that HIF1 alpha and CITED2 both interact with TAS1 with an affinity of 10 nanomolar by a range of different biophysical measurements. And so we have two disordered proteins that bind to a partially overlapping binding site on their shared folded binding partner with equal affinity. And so we decided that we really wanted to understand at the atomic resolution level how HIF1 alpha and CITED2 were interacting with TAS1 and whether perhaps both proteins are able to interact with TAS1 at the same time due to their partially overlapping binding site, and if this might play a role in allowing them to be very effective competitors for binding of the TAS1 domain. So we decided to use our favorite method, which is solution NMR spectroscopy. So here I'm showing you a selected region of the HSQC spectrum for N15 labeled TAS1, where each of these resonances corresponds to an amide backbone peak or region in the protein primary structure. And I've labeled the amino acids giving rise to these signals in this diagram. And simply what we were trying to do in this experiment was we wanted to be able to identify which residues of TAS1 interact with HIF1 alpha, which residues of TAS1 interact with CITED2, and perhaps if there are any residues of TAS1 that in the presence of a mixture of HIF1 alpha and CITED2 might be sensitive to binding of both. And so here I've shown the representative spectrum in black of N15 TAS1 bound to 1 molar equivalent of HIF1 alpha. And in order for us to discriminate between residues that interact with HIF1 alpha or CITED2, we need for the CITED2 bound spectrum to be sufficiently different. And so that's exactly what we observe here. So I've now superimposed the blue spectrum, which is N15 TAS1 bound to CITED2. And so we can see that most of the resonances are in different positions in the presence of CITED2 relative to HIF1 alpha. So this allows us to easily determine which parts of TAS1 are bound to HIF and which parts of TAS1 are bound to CITED. And so then we did our big experiment, which was to mix a 1-to-1-to-1 mixture of N15 TAS1, unlabeled HIF1 alpha, and unlabeled CITED2. And what we observed was quite surprising. And so this is the red spectrum shown superimposed on the HIF and CITED representative spectra or reference spectra. And what we saw was that we only observed resonances for this mixture of all three proteins that corresponded to the TAS1 CITED2 complex. And so what this implies is that CITED2 is able to fully displace HIF1 alpha from TAS1 at equimolar concentration. And this did not agree at all with our expectation based on the affinities that we had measured, because we had measured that HIF1 alpha and CITED2 have equal affinity for binding to TAS1. So if anything, we would have expected to see a mixture of some resonances corresponding to TAS1 in the HIF1 alpha bound state and TAS1 in the CITED2 bound state. And so this was quite surprising. It was an apparent violation of the laws of thermodynamics as we understand them. And so we needed to develop some new assays to understand this further. Simply, one of the limitations of this experiment was that we are doing these experiments by NMR, and we require high concentrations of protein for our samples. And we really wanted to understand what would happen to this behavior if we were to vary the HIF1 alpha and Cyto2 concentrations in a wide manner. But we really don't have access to a wide range of concentrations by NMR. And so we decided to develop another assay, which was to look at the competition process using another biophysical measurement, fluorescence anisotropy. And so in these experiments, we set things up in a slightly different way, just because there are some considerations for developing anisotropy or polarization measurements. And so in these experiments, what we did was we started with a fluorescently labeled HIF1 alpha peptide that we could then form a complex with TAS1, and this would result in a high anisotropy value. And then we can titrate in Cyto2 and look at the displacement of HIF1 alpha as a function of Cyto2 concentration. And this results in a decrease in the anisotropy that we measure due to the release of the fluorescently labeled HIF peptide. And we can then track that as a concentration dependence with Cyto2 added into our cuvette. And what we see is that instead of observing the expected affinity of Cyto2 binding to TAS1, which would be 10 nanomolar, we see this very distinct behavior in this experiment, where we have a very rapid drop-off in our anisotropy signal that corresponds to release of the fluorescent HIF peptide. And we measure a KD apparent from this measurement of 200 picomolar. So that's a 50-fold increase from what we would expect based on our other measurements. So this implies that there's something about having HIF1 alpha present that is making Cyto2 a better competitor. The other important piece of information from these experiments is this inflection point here, which corresponds exactly to the concentration of TAS1 that's present in the cuvette. So what this tells us is, once again, using a whole different set of experiments at a very different, in a very different concentration regime, that once again, we've observed Cyto2 being able to fully sequester all of the TAS1 from HIF1 alpha. But this 50-fold difference in KD still isn't enough to explain the odd behavior that we saw by NMR. And so we decided to change around how we were doing this experiment simply by starting with labeled Cyto2 protein and then competing with HIF1 alpha. And this is where we really started to understand what might be happening, because we saw an apparent unidirectionality in these experiments, where if we try to add in HIF1 alpha to displace the pre-bound Cyto2 from its complex with TAS1, what we see is that we require very, very high concentrations of HIF1 alpha to do so. And we can measure a KD apparent now that is shifted from the expected 10 nanomolar all the way to 900 nanomolar. And so this really reveals that HIF1 alpha is easily displaced by Cyto2, but once Cyto2 is bound to TAS1, HIF1 alpha is not as easily able to displace it. And so this was really surprising and of course made us very curious about what was happening, but really where we started to get some insight as to what was happening, because of course, you know, we had this idea that HIF1 alpha and Cyto2 were able to bind to TAS1 and form perhaps a ternary complex that might be able to accelerate dissociation of HIF1 alpha. And so we decided to do some kinetic measurements to understand whether there were differences in the rates of HIF1 alpha and Cyto2 either associating or dissociating with TAS1. And we decided to do them in a competition format. And so what I'm showing you here is, again, we're using a labeled HIF1 alpha peptide. We preform a complex with TAS1, and then we can compete with excess HIF1 alpha or Cyto2 and look at the release of the HIF peptide. These are now intensity measurements. So what we look at is we can see an increase in intensity as we release HIF1 alpha from the complex. And when we do these experiments competing with HIF1 alpha or Cyto2, we see very different behavior. So on the left, we're showing stop flow measurements that are carried out with a dead time of 2 milliseconds and over a span of 10 seconds. And what we can see is that we see this nice exponential increase in signal for Cyto2 displacement of HIF1 alpha, but HIF1 alpha displacing itself occurs very, very slowly. And it turns out that we actually have to do a manual mixing experiment with a dead time that's equivalent to the exact time of the stop flow measurement, so 10 seconds. And it goes over over a span of almost 200 seconds to see that same exponential increase in the HIF-1 alpha signal when HIF is competing against itself. But it's worth pointing out that in these manual mixing experiments, the Cyted2 has already fully displaced HIF-1 alpha from the complex. And so Cyted2 is able to rapidly displace HIF-1 alpha, whereas HIF-1 alpha is following a more normal process of displacement in these measurements. And what was most informative about these measurements is that we could do them at a range of HIF-1 alpha and Cyted2 competitor concentrations. And what we were able to observe from these measurements is that while HIF-1 alpha was able to displace itself in a concentration independent manner, we saw that the rate or the observed rate constant that we were able to measure for dissociation or what we hoped we were measuring as dissociation of HIF-1 alpha in the presence of competing Cyted2 was actually not a concentration independent value, which is what we would expect for a dissociation measurement, but in fact, it was a concentration dependent value that increased increase in a proportional fashion to the Cyted2 concentration. And what this tells us about the mechanism is that we are looking at something that we can call an associative mechanism. So what that means is that Cyted2 is binding to the HIF-TAZ1 complex before HIF-1 alpha dissociates in these measurements. And so this was a really nice confirmation of our original hypothesis, which was simply that Cyted2 might be able to bind to the HIF-TAZ complex before HIF needs to associate or dissociate, and that might be key to its ability to function as a really potent negative regulator of HIF-mediated transcription. And so, of course, as structural biologists, we wanted to understand how this might be happening. And so we went back to some of our favorite NMR methods, in this case, looking at the labeled HIF-1 alpha and Cyted2 peptides in complex with TAZ1. And we were able to use fast timescale dynamics or spin relaxation measurements to characterize the dynamics of the HIF-1 alpha and the Cyted2 peptides in their TAZ1 complex. And so here I'm showing the results of these analyses, which are shown as order parameters that are calculated by the spin relaxation rate constants that we measure. And what happens in these order parameter values is simply that anything that has a higher value, so around 0.8 or higher, is assumed to be rigid in a complex with TAZ1, and any of the residues that have lower order parameter values are assumed to be flexible in their complex with TAZ1. And so what we observed was that for HIF-1 alpha, despite the fact that we saw 3 helices forming in the structures, what we could observe is that HIF-1 alpha had 2 helices that seemed to be adopting a relatively rigid conformation in complex with TAZ1. But there was an N-terminal helix, termed the alpha A helix, that is actually quite dynamic in its complex with TAZ1. And the reason that this was most notable is actually that alpha A, as I'm showing here on the right-hand side, occupies a partially overlapping region of TAZ1 that Cyted2 alpha A would also need to interact with. And it turns out that once Cyted2 is bound to TAZ1, its alpha A region, and in fact, most of the polypeptide, is quite rigid in its complex with TAZ1 with high order parameter values. And so this suggests that there are residual dynamics in the HIF-1 alpha bound state of TAZ1 that are allowing for potential competition by Cyted2, perhaps in this region. And so we decided to use further NMR experiments to characterize that, that potential process where this region at the N-terminus of Cyted2, the alpha A helix, may be able to bind to the TAZ1 HIF-1 alpha complex. And so to do that, we knew that if we worked with the full-length peptide, we were only going to observe the Cyted2 TAZ1 bound state. And so we decided to make a truncation peptide where we truncated everything C-terminal to the LPEL, or C-terminal to the N-terminal region of Cyted2. So we removed the LPEL binding motif as well as the C-terminus. And what we were able to observe is that if we did NMR titrations with this truncated peptide, we now saw a very different behavior. And I'll come back to what these titration data actually tell us later in the presentation. But if we were simply to map which residues of TAZ1 were affected by binding of this truncated Cyted2 peptide, what we were able to observe is that all of the residues localized to the region of TAZ1 where Cyted2 alpha A would be expected to bind. And so what this does tell us is that HIF-1 alpha remains in its complex with TAS1, but it is sensitive to binding of CITED-2, and particularly that N-terminal region that's perhaps allowing for formation of a ternary complex. So this was really structurally quite informative, and we were able to look at the structure of the N-terminal region of CITED-2 that was included in that truncation peptide. And we were also able to infer from the structures of HIF-1 alpha that perhaps we could form a peptide in a single polypeptide chain, so make a chimeric fusion peptide that included regions of HIF-1 alpha and regions of CITED-2 that would be occupying non-overlapping binding sites on TAS1, and that perhaps this might allow us to model the ternary complex. And so this is in fact what we were able to do. We made a fusion peptide. This is work that was done by Frank Appling, who is a postdoc in Peter and Jane's lab. And Frank was able to make a fusion peptide where he fused the N-terminal helix and LPEL motif of CITED-2 to the alpha B and alpha C helices that are in the C-terminal region of the HIF disordered transactivation domain. And he was actually able, much to our surprise, to solve a crystal structure of 2 angstrom resolution of the fusion peptide bound to TAS1. And what was most informative about this structure was that the corresponding motifs in the fusion peptide bound in almost the exact location of those motifs in the solution structures of CITED-2 or HIF-1 alpha bound to TAS1. And so this was quite exciting. We finally had what we thought was a very nice model of the ternary complex formed between HIF-1 alpha and CITED-2 with TAS1. But then as Frank started doing more NMR experiments on this fusion peptide, he stumbled across something pretty interesting that wound up being very informative for understanding how HIF-1 alpha and CITED-2 compete. And that's simply that in solution, he found that binding of the HIF C-terminal helix and the CITED N-terminal helix, so HIF alpha C and CITED alpha A, is actually mutually exclusive. And there's a molecular tug of war that's happening between these two motifs. And what this tells us is that if CITED alpha A is bound, it disfavors binding of HIF alpha C. And so this could be really critical to understanding how CITED is able to then drive the dissociation or facilitate dissociation of HIF from the TAS1 complex. And so, of course, we were still very interested in understanding what makes CITED-2 such an effective competitor. And so we made some more constructs. So I've already told you about this N-terminal truncation construct where we were looking at just the role of the CITED-2 N-terminal region in driving competition and interactions with the TAS1 HIF-1 alpha complex. But we decided to make a range of different peptides that we could use as tools for understanding how the different regions of the CITED-2 peptide are able to mediate competition against HIF-1 alpha. And so we decided to make a mutation where we mutated the non-proline residues of the conserved LPEL motif to alanines. And we also made another truncation construct where we truncated C-terminal to the LPEL motif so that we would then be looking at the effect of losing just the C-terminal region that we call phi C. And we were able to do our same anisotropy competition measurement where we are looking at the ability of these CITED-2 peptides to compete against HIF-1 alpha. And what we can observe is that anytime we start taking away a key binding region, we are now increasing the affinity or the KD apparent for competition in this system. And so this tells us that there's a lot of interactions between the different different CITED-2 binding motifs, and that the interactions or the synergy between the CITED-2 binding motifs within the polypeptide chain are really important for allowing CITED-2 to be such an effective competitor. And so this was very exciting, and we decided to then look at all of these different constructs by NMR. And so these are data I've already showed you. So this is simply the data from the beginning of the presentation where we can see that if we have the full length CITED-2 peptide, it is only observed in complex with TAS1, even in the presence of an equal amount of HIF-1 alpha. And we decided to do those same competition experiments now with each of our truncated peptides. And so these are the data I told you I would come back to. With the N-terminal truncation, where we only have the alpha A region, and we see a nice pattern of chemical shift changes upon increasing concentrations of CITED-2. And we can do these experiments for all of these peptides. And what we observe is that we actually see some commonalities between the titrations. The commonality is that the pattern of chemical shift changes we see from the HIF-bound state to the cited-bound state is actually conserved between all of these different peptides. And that conservation of chemical shift behavior tells us that there is likely something similar between all of these. And if we look just at this diagram or schematic of all the different peptides that we're working with, the only thing that all of these constructs have in common is this N-terminal alpha A containing region. So that tells us that something about that N-terminal region is important for binding and competition with all of the peptides that we're testing here. The other thing that we can see is that we're seeing an increase in the population of the cited 2-bound state for peptides that are better binders and competitors. And so that doesn't seem too surprising. And so we saw all of these NMR data, and we realized that these were pretty featureful and could probably tell us a lot about the process. And simply what we were interested in understanding further, as another confirmation of our prior work, was simply whether these data and this complex behavior could be consistent with the 2-state model that I'm showing here on the top of the slide, where HIF-1 alpha would need to dissociate before cited 2 could bind to TAS1, or if these data are more consistent with a perhaps 3-state or higher order model, where we could form a ternary complex between TAS1, HIF-1 alpha, and cited 2 that then would allow HIF-1 alpha to dissociate once cited 2 is already bound. And so to discriminate between these two limiting and really minimal models that we're showing here, we decided to use a method called NMR line shape analysis, and specifically 2D NMR line shape analysis. And what this allowed us to do is simply question whether the limiting models that I just showed you, the 2-state or the 3-state model, would allow us to reconstruct the NMR data from our titrations with the different cited 2 peptides. And so here I'm showing example data for the titration with the cited 2 APAA peptide. What we're able to observe is that these are experimental data here on the left. I think it's pretty obvious that this 2-state model cannot recapitulate what we observe experimentally. But if we allow ourselves to use the 3-state model and invoke the ternary complex, we're now able to model the experimental data almost perfectly. And so this was a really nice confirmation, once again, of the ability of cited 2 to bind to HIF-1 alpha and form a, or bind to TAS1 already bound to HIF-1 alpha and form a ternary complex. And so the final piece of the puzzle understanding here, now I hope I've convinced you that we understand a lot about the properties of cited 2 that allow it to compete with HIF-1 alpha so effectively for binding of TAS1. But for a long time, we also neglected the properties of TAS1 that might be allowing for this behavior to occur. And this is simply just due to some common perceptions in the field and also in the literature that the folded domains of CBP and P300, like TAS1, are thought to be rigid scaffolds for binding of different disordered ligands. And that would really be the simple understanding of, if the folded domain is really just there as a passive partner for interacting with all of the different disordered proteins, that would remove a level of complexity. However, as we worked on this project, we started to appreciate that there is no level of complexity that cited 2 seems to have left unturned in terms of competing with HIF-1 alpha. And so we started to look at TAS1 and its behaviors, and specifically not only its structure, but also its conformational dynamics. And so what we hypothesized, just looking at the solution structures of TAS1, which I've now just colored in the same scheme that I've used for HIF-1 alpha in orange and cited 2 bound TAS1 in blue, was we hypothesized that perhaps there was something about the TAS1 conformation that was suitable for cited 2 binding when HIF-1 alpha was bound, but that once cited 2 was able to bind, it would make HIF-1 alpha binding structurally and or energetically unfavorable. And so to understand this further, we once again turn to our NMR relaxation measurements. And these are those same order parameters that I showed you earlier for the HIF-1 alpha and cited 2 bound to TAS1. But now we're looking at TAS1 in these experiments. And while there's a lot of data here that I don't expect you to be able to digest, I hope what you can appreciate is simply that the red points here that correspond to TAS1 in its free state without any disordered protein binding partner, the red points are uniformly And so that implies that TAS1 is more flexible in its unliganded form than when it's bound to either HIF1 alpha, which is shown here in black, or CITED2, which is shown in blue. And the other thing that we were able to identify is actually if we just subtracted the order parameters for the HIF1 alpha complex from those of the CITED2 complex, what we saw was that the difference was almost always positive. And what that tells us is that TAS1 is more rigid when it's bound to CITED2. So simply that binding of CITED2 is rigidifying the structure of the TAS1. And that could be an origin of why HIF1 alpha is no longer able to recognize the complex so effectively. And we can map where most of these changes occur. So these changes in order parameter from the previous slide. And what we can see is that they actually link the binding sites of the CITED2 alpha A helix and the alpha B and alpha C helices of HIF1 alpha on the other side of the molecule. So this is really a nice structural and dynamic evidence for allostery in the system, where CITED2 is able to act as an allosteric effector for dissociation of HIF1 alpha. And so overall, we've developed this mechanism for understanding how HIF1 alpha is displaced by CITED2. We can identify that CITED2 is able to compete in a nearly unidirectional or apparently irreversible fashion, and that simply for HIF1 alpha to reverse this process, there needs to be a much higher accumulation of HIF1 alpha than CITED2. We know that this process is really dependent on disorder in the HIF1 alpha and CITED2 complexes, but also on the dynamics and the allosteric conformational changes in TAS1. And now with the last few minutes, I'd just like to talk about some other work that we've done now in understanding how this has, what the implications of this work is for biological function. And so simply what I've already told you is that we've identified that the CITED2 peptide we've worked with in our biophysical experiments is a very potent negative regulator of HIF-mediated transcription. And the HIF transcription factors are associated with a number of diseases such as cancer and cardiovascular disease, and also with diseases that are associated with vision loss. And so we were really fortunate to be able to start a collaboration that's still ongoing with our colleague Marty Friedlander at the Scripps Research and his postdoc Ayumi Ouchi, who allowed us to explore using different animal models for understanding the role of the CITED2 peptide in modulating HIF activity in known ischemic retinopathies. And so while I don't have a lot of time to go into the details of this model, but I'd be happy to answer any questions about it in the Q&A. What we've been able to do is use this common murine model called the oxygen-induced retinopathy model to characterize the ability of CITED2 to inhibit HIF-mediated transcription in vivo. And so in this model, we're able to use or exploit the property of the mouse in that the mouse retinal vasculature does not develop until until 3 weeks after birth. And that's a really hypoxia-driven process. And so we can actually interfere with that process by mediating the amount of oxygen that's available to the mouse. And by doing that, we're able to then control when the hypoxic stimulus happens that will allow for a growth of new vasculature. And we can actually do that in a way that it would allow us to model the phenotypes that would be associated with ischemic retinopathies in human patients. And so these data here shown on the bottom are simply images of retinal tissue flat mounts where we've stained for the vasculature. And so here at P17, which is the endpoint of our model, we see the very characteristic behavior in this model where we have formation of pathological vessels that we call neovascular tufts, and the central region where the mouse is actually not producing any vessels due to the start of the model where we provide excess oxygen. And so we wanted to understand if CITED2 as a potential inhibitor of HIF1 alpha, and as I haven't mentioned yet, I should mention that HIF1 alpha controls the expression of a number of known pro-angiogenic cytokines that are known to contribute to the phenotypes in this model. And so we were really interested if we could use this model to understand how CITED2 might be interfering with HIF activity in the retina. And so, of course, we started with our buffer control. This is simply our vehicle control where we see the characteristic phenotype. And then we treated the mouse at the same, using the same protocol with the CITED2 peptide. And what we were able to observe is that we see near normal vascular patterning of the retinal vessels after treatment with Cytid-2, which is allowing us to determine that Cytid-2 is actually very effectively competing with HIF-1 alpha in this model. And then our collaborators suggested that we needed a negative control. And we did these experiments many years ago. And so we decided that our negative control would be this Cytid APAA peptide, where we mutated the non-proline residues of our conserved LPEL motif to alanine. And that indeed functioned as a negative control in our experiment. We could see that we can quantify the formation of vessels as well as the area of vaso-obliteration using a deep learning segmentation algorithm. And what we're able to observe is that the Cytid-2 peptide at all the doses we tested is a really potent rescuer of both the neovascular and vaso-obliteration responses in this model. But the Cytid APAA peptide does not as effectively do so. And we can also understand that the Cytid-2 peptide is able to downregulate transcription of known HIF target genes in our model. So it tells us not only that Cytid-2 is able to effectively rescue the model, but it's doing so by the mechanisms we expect. And we can see that our negative control, the APAA peptide, is not able to do so. And so at this point, I hope that I didn't rush through the data too much and that you were able to grasp the idea that this APAA peptide, which we use for some of our biophysical measurements, actually isn't a really good negative control as we think about negative controls, because we were able to observe in our competition experiment that it's still able to compete with a KD apparent of 50 nanomolar. And anybody that's ever really looked at inhibitor development, 50 nanomolar is still pretty good. And so we were really curious why it functioned as a negative control in our experiment, since we think that, it's still doing a pretty good job of competing with HIF-1 alpha. And the secret came from then going back to our anisotropy competition experiment. So I showed you earlier that if we start with the Cytid peptide with no mutations, we see that it requires a very high concentration of HIF-1 alpha to displace it, and we're able to measure this change in the KD apparent from the expected value of 10 nanomolar to 900 nanomolar. However, if we do that same experiment, but now we label the Cytid APAA peptide with our fluorophore and compete with HIF-1 alpha, we now get back the KD apparent of 10 nanomolar. So what this tells us is that HIF-1 alpha is able to rebind to the Cytid-2 APAA complex in a way that HIF-1 alpha cannot do so when the APAA mutation is not present. And so this is now a fully reversible competition process, which explains why in the ischemic or hypoxic retina, Cytid APAA is a very poor competitor and it does not have an effect in the OIR model. And so I hope today I've been able to share our results describing how Cytid-2 is able to potently displace HIF-1 alpha from its complex with TAS1 by a unidirectional switch-like mechanism. And that we think that this really allows us a new insight into how disordered proteins like Cytid-2 might be able to be used in a therapeutic context, specifically through the work that I presented at the end of the talk on understanding how Cytid-2 can mediate HIF-driven angiogenesis. And with that, I'd like to thank everybody that contributed to this work. So of course, most of this work was done when I was still a postdoc and staff scientist with Peter Wright and Jane Dyson, as well as working with Marty Friedlander. Ayumi made critical contributions to the animal work with the oxygen-induced retinopathy model, testing the Cytid-2 peptides, and Frank did the outstanding structural characterization of the chimeric fusion peptide. And with that, I'd just like to make a plug. As Chi mentioned in his introduction, I have just joined the UNC Department of Biochemistry and Biophysics here at the medical school. This is a really beautiful place. This picture barely does it justice. And we're currently recruiting at all levels. So I'd really be, if anybody is interested in joining us to work on projects involving disordered proteins or protein dynamics, as well as peptide therapeutic development, I'd be happy to speak with you further. And I look forward to your questions. Thank you so much.
Hi, Rebecca. It's I'll be moderating the questions for you.
Great.
Because she had to leave a little bit earlier,
yeah.
So if you don't mind. And we have a couple of questions already, and for the sake of time, We will answer them, you will answer them, and then we will go with the second speaker. And if you don't mind staying, if any other people from the audience have more questions, so we can address them. So the first question is coming from Ryan J. Torres. Great talk. I'm in the MAP lab at U of M. I have been studying similar things. What fluorophore was used in the kinetic experiments, and where was it placed on the peptides?
Sure. Hi, Ryan. So we used two different fluorophores depending on the experiments we were doing, but we of course did all the necessary controls to make sure that they didn't have an effect. So we were using the Alexafluor 488 and 594. And they were placed in different, so on the HIF1 alpha peptide, we used a mutation that's been described in the literature not to have an effect because HIF1 alpha actually has two native cysteines, one that's near the N terminus and one that is in a binding region near the alpha B helix of HIF1 alpha. And so we actually introduced a C800B mutation at that point so that we're able to label near the N terminus of the site of the HIF1 alpha peptide. And that was our labeling site for those experiments.
Thank you so much. And I'm going to go with the second question, which is coming from Christian. Hi, Rebecca. When you showed the KDs of displacement of HIF1 alpha with cited and vice versa, you had these hysteresis. Is that due to the fact that the titrations were done too fast, such that the kinetic effects may be effects play a role? Or alternatively, can you, by inclusion of the appropriate concentration of the tertiary complex, explain all data naturally with a 3D stage model? There are more questions, but let's go with these two alternatives and we'll go with.
Okay, so to address the possibility of kinetic effects, so we did explore that, and so we did a number of controls where after these were all done on separate samples where we would prepare the relevant mixtures, and that would allow us then to monitor changes or potential changes over time. So we can conclude that everything here was at equilibrium. So there's not necessarily a kinetic effect on the titration behavior that we observed. But so what we think really is just, it explains, so what I haven't had a chance to really delve into simply just because this is certainly challenging and would require someone to have a lot more time to dedicate to this than I had, would simply just be to understand what the kinetic steps are that lead to formation of the ternary complex and also of exchange of the ternary complex to the cited bound state. And simply what we think is happening is that we know that the binding of disordered proteins in general is relatively, we like to think of it as something that can happen fast and then, something that's close to a diffusion limited process. And we've done some association experiments by stop flow that identify that there are fast processes that occur on the order of 10 to the 7 per second. And so, I think that what we're observing and, getting to the second parts of your question necessarily that Magda didn't read out, which is, understanding what the KD of the ternary complex might be. These are all outstanding questions that we still have really to dissect the individual steps of the process and understand what the individual rate constants would be. We hope that we might be able to determine those from the NMR line shape analysis that we carried out, but we simply just don't have enough points to really refine those parameters. So we have some estimates and those are presented in the supplement of our recent PNAS paper, which really describes the fact that we know that we have a fast process that reflects formation of the ternary complex by cited 2 binding to HIF-TAZ1, and then a slower process that represents the ternary complex falling apart, so HIF dissociating. But we don't necessarily know whether that's really sufficient. And coming to your last question, so Christian has asked whether 1 ternary complex is sufficient for describing this, and that we don't know. But we think that the way we've set up our experiments so far, we were blind to understanding whether there are, two interconverting or more interconverting complexes that could be the ternary complex.
One more question before we go to the second talk. It's from an anonymous attendee. Great talk. One question: have you studied TAS1 HIF1 alpha in presence of DNA? If yes, does DNA has an effect on TAS1 HIF1 alpha interaction?
So we have not done those experiments simply because we are working with proteins that are actually just regions so in this case, we're working with just the C-terminal disordered region of HIF1 alpha, which does not on its own have any DNA binding capability. The DNA binding regions of HIF1 alpha are at a different part of the sequence that we do not have in our constructs. So we don't have any way in these experiments to test what the effect of, or expect that there would be any measurable effect in these measurements of the DNA in these types of experiments.
Thank you so much, and thank you for your great talk. And now I'm passing it to Varun to introduce our second speaker. Yeah, it's your turn now.
Yeah, thank you, Magda. All right, I see Dr. Yadav. So first, my introduction. I'm Varun Govind. I'm a professor of radiology at the University of Miami, Florida. Some of my current human research activities involve probiotic nutritional supplement interventions and subsequently measuring the influence of these supplements on brain health using various methods. It's because of this research background of mine, Dr. Rams has asked me to introduce the next speaker, Dr. Hariom Yadav. Dr. Yadav is an associate professor in the Department of Neurosurgery and Brain Repair and director of Center for Microbiome Research at the University of South Florida in Florida. His current research topics are focused on understanding how gut microbiome abnormalities impact brain functions such as learning and memory. As part of this focus, Dr. Yadav's team has come up with several prebiotics, probiotics, synbiotics, and postbiotics with a goal of modulating gut microbiome composition and diversity in such a way to reduce inflammation and perhaps leakiness of the gut, specifically in older adults. Dr. Yadav's team has initiated a new study titled Microbiome in Aging Gut and Brain. As we can see this as the background of Dr. Yadav's image. And his team initiated this new study some time ago. And the aim of this study, as I understand, is to investigate gut microbiome alterations in the aging gut and brain. Dr. Yadav uses personalized nutrition and microbiome approaches in preclinical and clinical studies. And his research is supported by the US federal sources such as NIH, Department of Defense, and National Science Foundation. So far, Dr. Yadav has published more than 130 peer-reviewed articles and earned several national and international awards for his research achievements. And I was told that he also commercializes the microbiome technologies developed in his lab, and perhaps some of the microbiome products developed in his labs too. The title of his talk today is The Role of Microbiome and its Modulation in Maintaining Our Brain Health. Dr. Yadav, it's a great pleasure to welcome you, and the Zoom podium is yours now.
Great. No, thanks a lot, Govind, for introducing me. And it's nice to meet you, everyone. Let me just pull out my PPTs and blow up them here. Can you see my slides here?
Yes, I can see.
Great. So thanks a lot again for inviting me here, specifically for Dr. Rams for giving me this opportunity to present over some of the research. I'm sorry, I think so I need to go on the first slide. This is my last slide. Hold on. Something else is happening. Sorry for that. So thanks a lot for everyone to be here, and I would like to introduce some of the research what we have been doing in our lab, and we'll be happy to discuss further. So as Dr. Govind has mentioned, is I'm going to talk mainly about the role of microbiome and its modulators in maintaining our brain health in here. Just to disclose, because my university asks always to disclose where I'm involved in other than the university services. So I'm the co-founder and scientific research officer of Post Biotech Inc., which is a startup is commercializing some of our technologies. And I'm also the consultant in the Must Be LLC to guide some of the technology in there. Before going in the much more in depth what we have been doing and that really what the other things are happening in this gut-brain axis is, there are some fun facts I would like to introduce with you here is, as we can say that actually based on the new research is we are more microbial than human. The reason is because we have 10 times more microbial cells than our own human cells. So in that way, you can think of as whether we are really more human or the more microbial. The second thing is we carry around 2 kilograms of the microbial mass of our total body weight. Although they are so tiny, they don't appear to be like that. But as I said, they are 10 times more, and then they make around the 2 kilograms of the total body weight. And then you might already know that is the Human Genome Project has taught us is, although the human genome is pretty long, but only express 23 to 24,000 genes. That means it makes only 23 to 24,000 proteins at any given time point, right? Whereas the microbial genome or microbes living inside or around our body actually makes more than 300,000 genes. So they make the more proteins than our own cells. Now, proteins are the functional unit of our each cell and the body, it controls the functions, right? So now you can again see that is who can control whom, whether the humans can control the microbiome or microbiome can control the human. However, this relationship is bidirectional, but you can see who can hijack the which system, right? Then coming back to the gut-brain axis, gut has as many as neurons as our brain has. For that reason, now it's coming more and more clear, we have second brain in our body. And that was not something I think, so we are trying to kind of more understanding more scientifically, but that knowledge has been for a long time. We have been talking about, oh, I have a gut feeling, right? But the feelings mainly are controlled by the brain. But then the new research is showing, yes, there is another brain which has the feelings on that, right? Another classical example is how the gut can influence the brain health is gut makes around 90% of the serotonin. And I don't think so I have to give this introduction to this group is how important the serotonin and the dopamine, which is produced by the 50% also, is contribute into the brain health or brain functions or brain, whatever the brain behaviors, you can say that, right? If you change these levels of these two neurotransmitters, you can completely change the whole, completely shift the whole personality of a person there, right? In addition to what the enteric nervous system produces these neurotransmitters, there are evidences showing that the microbiome also produces these neurotransmitters, including the GABA as well as the glutamine. So this is kind of the fun facts, knowing that is how the microbiome can influence our brain health and how important and why this is so important to be understanding further to maintain the better brain health, right? And this is another classical example of the communication happens between the gut and brain, and it happens differently in the healthy conditions versus the disease conditions. But these communications happens again in the both direction. The brain can influence the gut health, but gut health can influence the brain. And looking in the like more broader way is really this can happen in the one way is the direct wiring. So there is a vagus nerve which runs from the gut to the brain, or vice versa, both the efferent and afferent. They are directly connected by the wire. So it's like the very same system. Our computers can be connected with the LAN for the internet, whereas it can also be connected by the Wi-Fi, which is the humoral system or circulatory system, where the gut hormones or gut neurotransmitters can also go to the brain, but then brain hormones or the brain neurotransmitters can also come to the gut. So that's a like kind of the more Wi-Fi system. They do influence in the healthy condition. There is a different kind of the communication happens, but in the disease condition like the depression, anxiety, stress, or Alzheimer, or any other brain abnormalities, including the stroke or TBI, these communications take the different types. Another classical example is which we experience every day is the food intake cycle, right? Think about when we are hungry. Does anybody tells us, oh, you are hungry, go get the food? No. When we are hungry, the food seeking behavior starts from the body. But who initiates the food seeking behavior? Because our gut is empty, and gut tells to the brain, search food for me, right? Then once we start, we go and start searching food, get the food, and once we start eating the food, that's another thing is no one else tells us is, oh, you are full, you should stop. No, our automatic, the gut tells to the brain, we stop, right? And this cycle happens a couple of times in our day, right? Now, why this is important is because this gut-brain communication can alter the brain function in a couple of ways, right? For example, after eating this any kind of the food, it can make you happy, but it can also make you sleepy, right? So it also depends is the food you eat, how that react with your microbiome, and that can actually make the difference in how you will feel after eating, because you can pretty much see that the certain people after eating a food, they feel really happy, but other type of people actually they feel miserable about those foods, right? So this is the kind of the classical example how this gut-brain communication happens and make us feel also. The abnormalities in this whole food intake cycle or the gut-brain communication can also contribute into the progression of obesity or higher weight gain or the weight management. I will give you the very simple example. Let's say to maintain my normal body weight, in each meal, like 3 times of the meal, each meal I just eat the 30 bites. That's the calories. That's the calories I need in total of my day. Now think about if my gut and brain is not communicating, or these not communicating properly, or these communications are slow. What I might keep doing is just eating 2-3 bites extra because my communication between the gut and brain is slow. What will happen is, think about if each bite has, let's say, 20 calories. 2-3 bites means each meal I end up being eating 40 to 50 calories, or 40 to 60 calories, right? That means 3 meals per day. You can count 3 meals per day. Now, if I keep doing this for every day, per week, per month, and that's how the body weight progression can happen. So why these gut-brain communications are so important to managing our body weight is, this is kind of the classical example what happens, and the microbes living in the gut can influence these gut-brain communications. So that's kind of the very important axis. Now, thinking about the microbiome, really, the microbiome is kind of a very, very dynamic. So very plastic. It keeps changing throughout our life. So for example, when the babies are born, and then depends on how the babies are born, whether they are born by the C-section versus the natural birth, are going to have the different kind of microbial colonization, because the babies born with the natural death are going to acquire the mother's vaginal microbiome, whereas the babies born from the C-section are not going to have this. But on the same time, when the babies are born and now how they are fed, are they fed with the breast milk feeding or the formula feeding? That is going to differ their microbial composition. And then on the same time, when the babies or these kids grow and then switch from the liquid diet to the solid diet, there is a huge changes happens into the microbiome. Now, when we are growing as a toddler, depends on what kind of the other conditions, whether there is a antibiotic exposure, there is a malnutrition exposure, or the healthy are going to have the different kind of microbiome. Then when we are adult, that also depends on whether someone maintains their normal weight or become obese, or like having the other conditions, taking certain type of medications can change the microbiome. Also, the microbiome is different into the older people who are 65 years or old than the people who are younger, as well as the people who are living longer, more than 100 years. So centenarians has the different microbiome than the people who are in the middle age range. So you can see here what this cartoon is trying to show is the microbiome changes throughout the life, and the natural events can also change, but on the same time, unnatural events like the certain conditions which comes because of our health status can also change the microbiome and can have the impact in the longer term versus. So what we are trying to understand here is how the changes in the microbiome happens in the later life, and how they can be contribute into the maintaining the good brain health versus how they can also contribute into the progressing the age-related brain health conditions, right? Now, when we talk about the aging, the one thing is for sure is, first of all, aging is not a disease condition. So really the FDA approval for anti-aging drugs is, at least at that given time, is not possible. However, the aging is a single most risk factor for several human diseases, including cancer, lung disease, whatever like age-related or chronic diseases you talk about, they come with the aging, right? So with the older people, the risk goes so much high, right? We still don't understand why, right? But the age is one of the single most risk factor. However, the new hypothesis have been posed is called the zero science hypothesis. So to overcome is because the aging is not a disease, but it's still aging is so important to be understanding, as well as designing the strategies to reduce the aging related conditions, including the diabetes, obesity, Alzheimer, and other conditions, right? There have been the evidence is showing that is if we target the biology of aging, it can decrease the incidence of comorbidities, like for example, having the diabetes, obesity, and then other hypertension and the other conditions too, right? And that ultimately can increase the lifespan as well as the overall health span. And when we talk about the zero science hypothesis is based on the certain aging biology events, which is kind of macromolecular damage, which happens on the DNA damage kind of things, which when we get older, our cells goes through that. Epigenetics, inflammation, adaptation to the stress, proteostasis, stem cell regeneration, and metabolism, right? If we target these mechanisms, although these are fundamental mechanisms, most very important in each type of the cells, but there are certain ways to adapt them during the aging or during the old stage can actually can prevent the aging related conditions too. One of the most common one is the inflammation. So as we grow older, our body becomes just inflamed naturally. So inflammation is high, one way or another way. Now, when we talk about the gut or specifically the brain health, the people who has the higher systemic inflammation during their midlife, later in their life, they develop the more cognitive decline or the Alzheimer progress. And you can see here the number of the inflammatory markers who are high into the midlife stage, they actually has the higher cognitive score decline, they have the memory score decline, they also have the executive function and the language. These are all the mental health measures are significantly declined. There are also the other evidence shows that the inflammation not only present into the system or circulation, actually the brain also is inflamed in the midlife, also progresses the more Alzheimer and amyloid accumulation into the older age, and they develop the more dementia or the cognitive decline there too. The one important question comes is, okay, where this inflammation is coming from, right? Why the older people start getting inflammation? So although there are multiple theories, including the senescence, one of the emerging theory is this inflammation, which is basically the risk factor of several human diseases for the different organs, whether it's the nervous system, musculars, or the pulmonary, or the digestive cancer, or the cardiovascular problem. One of the single source is gut dysbiosis. Gut dysbiosis means the abnormalities in the microbiome. And how does this happen is when the gut dysbiosis increases, it increases the more inflammation in the local environment. That creates the more permeability in the gut, because otherwise the gut is very selectively absorbs the things what we eat, rest the things passed through the stool. But when this permeability increases, it allows the non-specific diffusion of the inflammatory signals from the gut lumen to the blood. And on the same time, there are bacteria and other microbes can also actually sneak out from there or leak out from there. And when they are exposed to the mucosal as well as the systemic immune system, that's how it creates the chronic inflammation. And this chronic inflammation creates the more age-related diseases. Now, looking back to the microbiome, how does the microbiome is different in the young versus the old people? And this is our new data showing that is, although the other people also have shown the microbiome is quite different into the old versus young. And this you can see here, the young people, definitely their microbiome clusters very differently than the older people. These are the classical differences in the major phyla happens in there. Is you can see the Firmicutes, which is a kind of a not good bacteria, are increased into the older gut. There are also the different kind of the other bacterial genera also propagates there. When we do the kind of the selective or the unique microbiota signatures, you can see here the young has some of the unique microbes which are not present into the elderly and vice versa. So based on the microbiome, you can also differentiate whether the this sample is or this microbiome is coming from the young people or the older people. So we are kind of trying to develop the biomarker based on that. Now coming back to the how does this gut permeability and leaky gut is linked with the age-related inflammation. So what we have seen, and I think so there are other papers also came out recently showing that is the older mouse has the higher gut permeability. And older people also has higher gut permeability, higher inflammation. The question comes is, does the microbiome induces that, or is a kind of some other sources? So we have done a direct experiment showing that is if we transplant the microbiome from young and from old into the young mouse. So these recipient mouse are young, but then we put the microbiome from the young mouse to young and young mouse to old mouse to the young mouse. What we have seen is when we put the old microbiome into the young mouse, they create some more gut permeability. And these are the other markers. These are systemic markers like the LBP and CD14 are the other markers of the gut permeability also increases, showing that is old microbiome actually lead to, or it's a causal to create the more gut permeability, which is linked with the higher inflammation. Why the old microbiome and how the old microbiome does it? One of the mechanisms what it happens is our gut has the very thick mucus layer. And the old microbiome actually breaks it. And how it breaks it? Because it suppresses the production of that mucus. So that glassy layer of the mucus is thin or it actually break down. And that's the reason why the leakiness happens here. And this is what we have seen exactly. When we transplant the older microbiome, actually the mucin producing cells decreases there. You can see here the goblet cell numbers significantly decreases when we transplant the old microbiome into the young, showing that is the older microbiome is causal to induce the leaky gut or the higher gut permeability by reducing the mucus layer, because the mucus producing cells are decreased in response to the older microbiome. Can be restored back by doing certain interventions based on the microbiome? The answer is yes. We have proven this by using a human origin heat-killed bacteria, which we call that as a postbiotic, D35, which reduces the aging-related conditions, including the leaky gut and inflammation. And this cartoon shows is the effect of this postbiotic or heat-killed bacteria on the most common mental health and the physical health related problems into the older people. So depression, anxiety, physical dysfunction, as well as dementia are most common to the older people. Now, when we feed these D35 or this postbiotic to the older mouse, which are around 80 weeks older, and which equates to the around more than 65 years old person. And what you can see here is when we do the open field test, which is a kind of the anxiety and depression behavior test, the mouse which are more less depressed and the less anxious, they are more exploratory. So they go more times in the center, which the green line shows that. As compared to the normal mouse or kind of the more depressed mouse or the anxious mouse, they will remain on the corner. So you can see here the D35 fed mouse really explore more into the center, and that is quantified by the distance traveled into the center as well as the time spent into the center. And then we also have done another test is which called the rearing test. So if you make some noise, the mouse will actually rear up. And these mouse which are fed on the D35 rear up less, showing that they are less anxious there. Another thing is to measure the physical function. We also have measured the ambulatory activity, total activity. Both were increasing to the D35 fed mouse, is showing that these mouse has the better physical function. And we can directly also test is the muscle strength. So there is a wire hanging test where we hang the mouse and then see how long they can actually hang. The longer they can hang, the longer means better the muscle strength they have. So you can see here the D35 fed mouse has the higher time to hang there, showing that the better muscle health, right? Now we can also challenge them for learning certain tasks. So you can also see here the D35 fed mouse learn much more quicker, showing that the whole learning and memory behavior was significantly increased by feeding this D35. And then we have published this story 2 years ago. And we also have seen that it decreases the leaky gut and inflammation, which actually contributes in the overall improvement of this aging related mental health condition. And this was a kind of the whole overall the story is why this D35 works is because the cell wall has a component called the lipotecoic acid, which is unique for this particular strand, which activates the TLR2 signaling, and the TLR2 signaling initiate the more mucus production in the goblet cell. That decreases the leaky gut, and that improves the aging related conditions by decreasing the learning and memory dysfunction, physical dysfunction, as well as the metabolic dysfunctions. And this is the product which we have transfer the technology and coming into the market soon as named as a Posibio. Another intervention we also have designed was the multi-strain cocktail by combining the 10 different strains of the human origin probiotics. And we also have conducted another study which was DoD funded recently, is to see if this 10 strain cocktail can reduce the Alzheimer progression into the Alzheimer mouse model, which is the APPS1. And you can see here when we feed this probiotic intervention, actually it decreases the cognitive dysfunction onset into the APPS mouse. It also decreases the A beta accumulation into the brain. Also, it decreases the inflammation into the brain, which is equates to the decrease the inflammation into the gut. I'm sorry for this last labeling. This is last two panels are the inflammation into the gut. So based on this data, what we have shown so far is the gut-brain communications are bidirectional and constantly happens throughout the life, which we can actually feel every day by the hunger and satiety axis. Microbiome is important modulators, and it's a dynamic. It keeps changing in each and every stage of our life. Aging is not a disease condition. However, manipulating the aging biology can reduce the comorbidities, which is the zero science hypothesis is based on. And inflammation is one of the important pillar of the zero science hypothesis, which is also a important risk factor for aging related disorders. And reducing the leaky gut can reduce the inflammation, which can really reduce the aging related mental health conditions, right? We are studying these different formulations. If anyone is interested, please feel free to contact us. We will be happy to collaborate for both preclinical and clinical studies there. Now, we recently also started to asking you a question is how microbiome can contribute to age-related brain health deterioration into the human as we grow older, right? So when we talk about in aging, actually cognitive decline, dementia, decline in the physical function, cancer, diabetes, anxiety, these are the age-related conditions. The question comes is, can we predict? Because when the dementia happens or when we go on the physical decline and reach to the wheelchair stage, really that time reversing back is hard. So is there any way we can predict early enough to slow down or prevent the progression of dementia and physical decline is so important there. To answer those questions, we have started a study called the Microbiome and Aging of Gut and Brain called the MIA-GB research study, which we are recruiting the people with the more than 60 years old. We are targeting like more than 500 people to be enrolled in this study at the 5 different sites. This is recently funded by the Florida Health Department, and we are recruiting. So we are leading from the USF, but we are also recruiting at the UCF, University of Central Florida, University of North Florida, Florida Atlantic University, Miami Juice, and it's kind of the first study focused on the microbiome and aging in the United States as well as in the world. Right. So this is the kind of the overall view of the study. Is we are doing this study in the 2 phase where we are recruiting the 4 different groups of the people, healthy, who are the 60 plus years old, plus the people who has the mild cognitive impairment, as well as the people who has diagnosed Alzheimer's disease or dementia. And then we also are recruiting the young healthy people to compare their microbiome at the baseline. So we want to develop the trajectories on the baseline to see how, based on these trajectories, whether we can differentiate who is healthy versus who is having the MCI versus who is having the dementia, young versus old, and then take that information, and we are also collecting the data and samples for the prospective longitudinal follow-up for 4 years, once in a year, for specifically the group of the healthy as well as the MCI, which is the mild cognitive impairment, early stage of the dementia, to see if we can predict who will have higher risk of developing the MCI or dementia in the later stage, and whether we can intervene there, right? So we already have quite good success in recruitment of these participants in different sites. We are collecting a lot of measures in here, whether this is demographic, exposomes, or cognitive assessments and physical assessment. We are also collecting the different biological samples, for example, stool, saliva, blood. We are also fractionating blood into the different kind of cell types to isolate there. One of the the analysis, the preliminary analysis we have done into these 4 cohorts, where you can see here the cognitive function declines into the older versus the young the smoker test looks pretty consistent. However, the people who has MCI versus the dementia, this score declines, and you can see in the other tests too. So these are our population based on the microbiome signature. Actually, we can see the certain trajectories where we can find out who is healthy. So we have the certain microbial trajectories which can show that this sample is coming from the healthy person, or this sample is coming from the person who has the MCI versus this person who has the dementia. So that's the kind of these trajectories. The red ones are showing the cognitive related trajectories, the health related trajectories. We can also differentiate them whether these samples are coming from the older people or these samples are coming from the younger people. This is a kind of a simplified presentation. What we are trying to show is, but this is the more algorithm based and the artificial intelligence based algorithm where we can find out these trajectories. So you give a blind sample, we can actually show is where this sample is coming from. So based on that, I hope I have shown enough evidence or data showing that is we launched a first of its kind multi-site study called the MIA-GV Consortium, which is harnessing to the power or potential of the microbiome-based biomarkers to predict the age-related cognitive decline and dementia progression. We are collecting the large data sets, which is available for the collaboration. So please feel free to contact us if you want to measure certain of your favorite markers in the either blood, in the saliva or stools, as well as the want to take a look on the other data. Our overall goal is to developing the markers to predict the age-related cognitive decline as well as the physical function decline. So both in the brain health as well as the muscle health. We are developing the precision microbiome signatures, which will be utilized to design the products for improving the personalized health, specifically the brain health. The take-home message for my talk is knowing your gut feelings better can keep our brain more healthy. So take care for your gut feelings, and that will take care for your brain health too when we are getting old. I would like to thank my team members, my collaborators, as well as the funding agencies for supporting our research. I'll be happy to take any questions you might have. Thank you.
Any questions from the audience? Okay, there is one question. Ashutosh Tiwari is asking this question. Gut microbiome also depends on lifestyle and food habit. Example, vegetarian, high meat based, or balance of both. How are you accounting for that?
Yeah, so that's a great question. We are collecting the 24-hour dietary recall, as well as we are collecting as much as possible. As I said, we are collecting the whole exposome, which defines the exposure of the body got throughout the life, which includes the diet, drinking habits, smoking habits, the different kind of exercise habits, and other lifestyle, including what kind of the jobs you have been doing, like all that matters there. So it's a really interesting question, and we will have that information to correlate with the microbiomes.
The next question is from Dr. Grisinger. So he says that I had a question regarding your error bars in the D35 experiment. I think he is referring to the postbiotic experiment. Mm-hmm. It seemed to me that the stars and the error bars did not really go well together. Could you or maybe go back to the slide and explain?
Sure. Let me bring the slides here. Okay, can you see my slides? Let's go back. Okay. So the question is the error bars here. So these error bars are plotted as a standard error of mean. And we had around 8 to 10 mouse in each group.
I see. Okay. Yeah, his question is, the stars and the asterisks and the error bars did not really go well together.
Yeah. So if we put this, convert this into the standard deviation, then yes, these error bars will be going definitely different. But because this is a standard error of mean, so there is a variation high, and these are always comes in. If you do any behavioral test, always your errors will be high. For that reason, your significance is going to be shrunken up. We plotted that as a standard error of mean, but that's what the p-value shows.
Okay, yeah, I understand. So what he plotted is SEM, standard error of the mean, not the standard deviation.
Correct.
That will be much higher. Okay, any other questions from the audience or the panelists? Okay, so, one question from me. So I think, I understand from your talk that when we grow older or when we age, the firmicutes is the one that increases in size as compared to the younger population. So, so what is that it produces? Neurotransmitter, is it the butyrate? My memory is saying that butyrate is the one that this particular group of bacteria has produced. Anything else that this group produces?
So no, that's a very interesting question. And let me give you a little bit kind of different aspect of this one is. What we are talking about when we talk about firmicutes, bacteroidetes, which makes actually around the 70 to 80% of the bacterial population in the gut. What we are talking about is a very big bird view, right? So we are looking from very far. We are really looking from 30,000 feet, right? Now, within the firmicutes, even like most of the probiotic, you talk about the bifidobacteria and lactobacilli, they belong to the firmicutes, right?
Yeah.
Now, but there are a lot of nasty bugs belongs to the those firmicutes too. Now, the overall abundance of firmicute is increasing because there are more probiotic or lactobacilli bifidobacteria, which is not true. But the firmicutes can be also accounted for increases because there are a lot more nasty bugs has been increased, which fortunately, unfortunately, whatever you say, they belongs to that particular phyla. But they increased based on the very bottom of the phylogeny is species or the genera level. But they account for the increase into the phyla levels, right? This classical example is not all the E. coli are the same. There are nasty E. coli who does a lot of infection, but there are E. coli as a probiotic too. So you can see that is the same E. coli differs on the strand level, and our technology is not as efficient right now to find out those strand level. And we are talking very up on the phyla level, because the phyla, then the order, class, then the family, then the genera, then the species comes, then the strand comes. And these all activity differs on the strand level rather than the phyla. So, but that gives a kind of overall bigger picture.
Okay, great. Thank you. And the next question is from Murugesan Velayudam. So when the gut is leaky, whether the microbiomes migrate or transfer into systemic circulation?
That's an excellent question. Remember that when I say leaky, does not mean there is a hole. The leaky means is there are loosening into the gut epithelial barriers, which allows whether it's a microbe or microbial ingredients, because the bacterial cells are dying every second. And when they disrupt, they can release the like LPS and the other kind of the components, which can also leak out, right? Which suppose not to leak out in the normal gut, right? So not always the microbial translocation happens, that the leaky gut allows the non-livable cells. However, if there is a more damage, then yes, the live bacteria can also leak out from there, and they can go into the systemic, and that can cause the sepsis problem too. And now there are more and more evidences are coming actually in the certain conditions, like for example, in obese, if the people has the higher obesity and the really severe obesity, they do have the live microbial colonization into their liver, which makes logical sense, is because think about anything we absorb from gut directly goes into the portal vein and goes the first organ in our body exposed by the whatever coming the blood circulation coming from the gut is the liver. So there are biofilms have been found into the liver of the people who has the severe obesity, and severe obesity has the higher leaky gut too.
Great. My final question is to bring the relevance of your talk to the talk series, that is Alzheimer's disease. So how do you connect, the microbiome or gut microbiome specifically to Alzheimer's disease?
So in the simplified way is, if there is a more leaky gut, it creates the systemic inflammation. It's not just the inflammation in the gut, which get translated or translocated into the brain too. There are new studies coming out is, if there is an abnormality in the microbiome, you create by either antibiotics or you transplant the abnormal microbiome to the normal mouse, that stimulate the microglia inflammation. So neuroinflammation, and the neuroinflammation triggers the higher formation and accumulation of the protein aggregates, whether it's amyloids or the tau, and then also creates the more neurodegeneration. So it's a kind of that you can just say that is a simplified version, but it's not that simple. But there are evidences saying is, if you change the gut microbiome, it can change your brain health.
Okay. Any other questions? Okay, if there are no questions, let's thank the second speaker, Magda. So are we going to bring the first speaker back?
I'm not seeing any questions for Rebecca, unless Rams and Daniil will have any questions. Some.
Great talks. Both the speakers really appreciate it very much. So sorry, I had to miss part of both the speeches. I got another meeting to go, so that's why Magda took over. Apologies for that, but I liked it very much. I do have questions maybe for another time I will ask. Danilo, you can ask questions. Yeah.
Okay, just a curiosity for Rebecca. I know, I understand that you performed HIF1 and CITED2 binding experiments by using fluorescence anisotropy, which is a brilliant, very sensitive technique. But I wonder, why not to use isothermal titration calorimetry, which is a very useful technique that does not need labeled proteins, and that, if performed correctly, can give you a lot of details in terms of thermodynamic parameters like entropy and entropy?
It's a great question. So I think the first answer is that we have. But we've done that for the full length HIF1 alpha and CITED2 peptides that we were working with. And because they are binding in the low nanomolar regime, ITC can be challenging just to find the right set of conditions to get reliable estimates of the thermodynamic parameters. Our reason for following through with the different constructs using the fluorescence anisotropy was simply that we had developed the assay and we had all the necessary reagents on hand. But I think that absolutely going back to some of those ITC measurements that we did a while back would absolutely be really beneficial. I think that there's a lot we can learn about the underlying thermodynamic behavior of these systems by doing those measurements.
Okay, thank you so much.
There is one new question from Pratima Mishra. This is to Dr. Yadav. What is the status of a healthy gut related to no disease?
Okay, so this is a great question. Don't know exactly what you want to know about this, but there are, as I said, the young people has the different microbiome than the older people. Although in generally, young people are considered to be healthy than the older people. However, it's not true always, right? But on the same time, if you look the microbiome in the centenarians versus if you compare the microbiome of the people who are 65 versus 65 to 80 years, their microbiome is different. In terms of the centenarians has the microbiome much more similar to the microbiome of the younger people, right? Now, because they had the different microbiome, that's how they are living longer. Or because they are living longer, that's why they have the different microbiome like young, is not. But the fact is the microbiome is pretty much go hand to hand the host health, because they always a bidirectional communication. If the body is healthy, it's going to feed the healthy ingredients to the microbiome, and microbiome is going to feed back the healthy ingredients to the body. If the body is sick, the classical example is if we are diabetic, normally we look for the blood glucose levels, but that does not mean that glucose is only high into the blood. It's everywhere. Every fluid of the body is high glucose. So think about how many juices or digestive secretions we pour into the gut. They all are bringing the more glucose. And that glucose will be fed to the microbiome too. And that means the bacteria which loves the glucose are going to thrive there. But the bacteria who don't like the glucose are going to either die or get deficient there. So it's kind of the simple explanation is it also depends on the host health, how the microbiome is going to be turns out. But on the same time, if you change the microbiome, it can also impact the host health. So it's a bidirectional communication. I hope that answers the question for her.
I think so, yeah, great. Thank you so much. So Dr. Rams, it's back to you.
Yeah, I think we're done. We're through. Thank you very much for the presentations, and thanks for hosting, guys. We have a break entire August, no ZOOMinar. See you in sometime in September, second week of September, I think. So watch out for the emails. Thank you all very much. Have a good summertime.
Yeah.
And best
Thank
wishes
you.
for
Thank you.
Thank you.
Thank
Thank you.
Thank you. Nice seeing you,
Bye.
everybody. Yeah.